All right, good morning, folks. So this handout, this worksheet, is to guide you through a virtual lab experience. So you're going to be using the computers to see what it's like to use a new genetic technology called DNA microarray. The link to get to this website is learngenetics.utah. But of course, to make it easy, you can just go ahead and check out Google Classroom, and I have the link right here for it. The title will be Microarray Technology. Microarray Technology. So you just click on the link for microarray. That'll be right here. And once this video is made, you'll probably find an attached link as well, just to know how to use the website. All right. So, in general, doing this activity has a purpose. The purpose is to learn more about microarray before we do it in class. So this is preparation. This is a pre-lab activity so that we can be better prepared and better understand the microarray lab when we do it in class together in our teams. So I hope you enjoy it. I hope you find it interesting. This is cutting edge technology, and this is helping people learn a great deal about how active or inactive their genes are, and how scientists and lab people and doctors are all using this technology to better understand the genes involved with other people's diseases and illnesses. So, let's see if we can't jump in. I'm going to show you kind of how to use the website, and then I'll let you free to just proceed, all right? So use the video as needed when you get confused. And if you don't get confused, great, just follow along the worksheet. But if you do hit some bumps in the road, of course, just stop the work on the worksheet and then watch the video for the section we're on, all right? All right, let's begin. First things first, this is exactly what you should look at when you click on the link. It says DNA microarray. You know you're in the right spot. However, if when you turn your computer on, you do not see this box, if you don't see it anywhere, that means the Flash player, which is the software this virtual lab runs on, it means it needs to be allowed or activated. So if you don't see this black box where you can click to begin on your computer, you need to go up to this little icon here. It'll look different for different web browsers, but you click here, and for Flash, you need to click Always Allow for this site. Now mine showed up, so it's already on Allow. But if your computer didn't show this, you need to click right here, and then reload the page. And then it will show up. And then you can start having some fun. Question number 1, it says, list past ways of studying genes. So let's do that. You click here, and then you just click on chapter 1. So you go one chapter at a time, right? So click on chapter 1, and you can see how visual it is. And it gives you this little information. It says, back in the day, a few years ago, scientists came up with many ways to study genes. So we used to study genes in different ways. So in the past, we used to map them. We would make mutations in DNA to study them. We'd clone DNA to study it. And we'd figure out the nucleotide sequences of them and the proteins they make. You can see, though, it says that just based on how many genes a human has, we're made of 20,000 different genes. Doing it that way, doing it the old way, one at a time, takes a very long time. And someone may dedicate their whole life just to study one of those 20,000. And still maybe not learn as much as they want. So far, you should have some information for question 1 and 2. Let's move on to question 3. It says, what is genomics and microarray? So we keep moving along with the chapters until we figure out that information. So when we click next, it gives us a chance to try to answer question 3. It says, wouldn't it be great if you could study more than one gene at a single time? So these DNA microarrays are really an impressive technology where instead of studying one gene at a time, you can get a whole organism's collection of genes or a bunch of their genes all at once and put their DNA and DNA for a specific gene, many of them, so you can fit thousands of genes on a little tiny microarray. It says genomics is a new word for a new science. It says genomics is a way to study many genes, thousands of them, for one organism all at once. So that's genomics. All right, the next question is, what is gene expression? And also relate gene expression and different body cells. Here we go, so let's check it out. Click next. And it shows you different cells. And it says to see powerful genomics, let's take a closer look at what makes various types of cells different. So gene expression. So it says, with a few exceptions, every cell in our body contains copies of about 20,000 different genes. It says different genes are turned on or off in different cell types. So gene expression means genes are turned on or off, which we know means they're making protein. And different cells, that say, have different genes on or off. That's what makes them different. All right, now let's answer question 6. All right, so question 6. All right, so here you go. So when a gene is on, it makes mRNA. We knew this because to make a protein, the gene first makes RNA, and then it makes other things after that. All right, so once the RNA is made, it then will make a specific protein from that RNA. So we know when the gene is on, it makes the RNA, and the RNA is used to build a protein, like these two proteins. These are the proteins of a muscle cell, and you can read here as you go through it. All right, question 7 are, what are the steps of making a microarray? It says, what do steps 2, 3, and 9 have in common, and how do you think this technology helps people? So let's click through the simulation and figure out what steps 2, 3, and 9 are, and what they have in common, and how this is a helpful technology. So we're going to click next. All right, this is showing you different genes are turned on or off in different cells to make them different. So each cell makes their own proteins to help them. And microarray allows you to study different cells and how all their genes are active or inactive at the same exact time, which is pretty cool. So here's all the steps. 1, 2. Every time you click, it'll give you another step. So we got step 2 and 3 here. 4, 5, 6, 7, 8, 9, and 10. Now, 2, 3, and 9 are supposed to have something in common, or we're supposed to read them and figure out what's in common with them. So, number 2 says, use massive amounts of computing power, technology, to figure out where all the genes are in your sequence. So, 2 is saying you can find all the genes along DNA using this technology, all the 20,000 different genes in a person. Number 3 says, is that you can make copies of every gene. So here is finding all the genes, here is making copies of all the genes, and then 9 says having a place for each gene in the microarray. So in the microarray, if you're going to study, thousands of genes at once, there needs to be a location in the microarray for each one. So I think we pretty much have our answer for that next question. So question 7, steps 2, 3, and 9 are about how we find all the genes, how we make copies of all of them, and then place them in an organized way in the microarray device. So now we're ready to do the next section of the worksheet, and that's to begin the simulation. Now I gotta be honest, as you do this activity in the simulation, you'll have to figure out how to click on different things to make the lab work. I'm just going to show you the very beginning part, otherwise it'd be a very boring video. But as you click on different things or ask friends next to you, you'll figure out how to get through the lab. Don't get stressed out if you don't always understand what a piece of technology is that you're using. Just try your best to follow along. Really, the idea is that you'll try your best to answer these questions of what's happening during the simulation. But you really just want to also get a sense of how amazingly complicated one of these labs is. If you had all the right technology and how much training you have to do it right. So let's begin the simulation and see what this virtual lab, what it's really like for someone in the lab to do this. So right now, we're going to figure out just what it takes to use one of these DNA microarrays. So we're going to select a new chapter, and we're going to go straight to the experiment. So this is the virtual lab. It says we're going to use the power of genomics and the microarray to study healthy cells and cancer cells. So we're going to look at how genes are being used in cancer cells versus healthy cells. And this is going to prepare us for our real lab in class. All right, so as we get a little background knowledge on the cancer cells, we use this information to help answer some of the first questions in the second section. So the first question is, the microarray contains multiple copies of what in each of its thousands of spots? Well, if you read up here and we've been following along, we realize that the microarray is used to study thousands of different genes, right? So we want to study different genes. And then it says, what question are we seeking evidence to explain using the microarray? Well, if you weren't paying attention, what we want to do is study what's going on with cancer versus healthy cells. What makes the gene use or the activity of genes in cancer cells different than healthy cells? If we know what genes are active in cancer cells, we know what genes to study. It says cancer is basically a disease where genes have gone bad. It says many genes control the way cells grow. When these genes stop working properly, like gene 3, it says it can lead to formation of tumors and cancer. Now, there's all different kinds of cancer, but it's all based on this idea where you have a certain gene, something happens wrong, and a bunch of cells start reproducing. All right, we're finally ready. So questions 4 through 7 are about doing the actual lab and doing the actual experiment. Questions 1, 2, and 3 let us know, look, we're trying to study cancer and what's different with the gene activity of cancer cells versus normal healthy cells. But then questions 4 through 7 are where we use technical equipment and try to figure out how to do the lab procedure itself. So you're going to click Next, and it's time for the experiment. These are all the different steps that you're going to do to perform the virtual lab that is doing a microarray. So check this out. This is all the equipment, and it's a lot. You'll see it adds up fast, okay? So this is all the real technologies a lab technician would use if they're studying the genes and how they're active in a healthy or sick person. Do you know what all these things are? No, of course not, and that's okay. Let's read the question, though. It says, once you start using the equipment, it says, first you collected tissue from cancer cells and normal cells. Then it said, second, you centrifuged or separated the mRNA from the rest of the stuff. It says, label the test tube below. So if you do the different procedures it guides you through, you'll end up seeing this, and you need to, on your worksheet, label the different sections and what they are. And the virtual lab will tell you what they are. And then you'll proceed following the instructions as you go, and in the end, you should understand what these colors mean for the different parts of a microarray, and you should be able to identify properly, using the virtual lab, what gene apparently is responsible for causing the cancer in your patient from this virtual lab. All right, so we click Next, and so we'll be using all this technical stuff. It says first, here's your patient. They have a tissue of skin cancer. And this just gives you an idea how to do the lab. They're always going to put something in front of you and use your mouse. You got to click on it. We're going to take it, and you drag it over. So you click and drag a lot. Click and drag. So from one person, you took a section of their skin that's healthy, and you take a section of their skin that looks like it's cancerous, okay? And then you look around, and you may not know exactly what to do, but you read the instructions, and you click Next when possible, or it'll want you to grab something here, okay? Right now, it wants you to click Next. So you can see how it kind of walks you through this lab. The solvent is a special chemical, and it's asking you to add this chemical to the healthy cells, so I'm doing that. Solvent is used for different things. Maybe it's protecting the cells. Maybe it's keeping them from getting destroyed. Maybe it's keeping something from happening. Maybe it's stopping molecular processes inside. I'm not sure either. We'd have to study it more. But the point is you follow the steps and realize all the technical equipment used. All right, let's click Next. So it won't let me go next. So what you have to do is you have to grab something. Let's see what it wants. So it wants you to grab the healthy cell, and you're shaking the materials. It says now place the cancer one. So you just follow the instructions. Now this video won't allow you to see the very end of the lab, but just continue doing these activities to finish the worksheet. All right, good luck following up and finishing questions 4 through 7.